This is a video abstract for the manuscript titled RNA binding protein DDX5 directs Tuft cell specification and function to regulate microbial repertoire and disease susceptibility in the intestine by Long et al. The intestine is home to a variety of different epithelial cells that work in conjunction to maintain intestinal homeostasis. One subset of intestinal epithelial cells are Tuft cells, which have diverse tissue-dependent functions. They protect the intestinal epithelium by reducing inflammation or warding off microbial infections. They have also been shown to act as viral reservoirs in the small intestine and have tumorigenic properties in the colon. In order for an intestinal progenitor cell to become a Tuft cell, the Rho GTPase CDC42 and the master transcription factor POU2F3 are needed. However, little is known about how these elements are regulated to drive Tuft cell specification. In the study, we investigated how these necessary elements are regulated in vivo by the dead box containing RNA binding protein DDX5. DDX5 is an RNA helicase that is a biomarker for cancer treatment resistance and has been shown to promote tumourigenesis in both the small intestine and the colon. But little is known about its role in Tuft cell differentiation and function. To assess the role of DDX5 in Tuft cell specification and function, we engineered an epithelial cell-specific DDX5 knockout mouse and compared it to a control using transcriptomic approaches. We found that in the small intestine, the DDX5 knockout mouse had a dramatic reduction in Tuft cell populations, found through staining for the Tuft cell marker DCLK1, as well as an altered susceptibility to ileal inflammation. In the colon, DDX5 knockout mice had altered microbial compositions and were protected against tumorigenesis. DDX5 was also found to alter Tuft cell function, as it promoted integrin signaling in antimicrobial responses, while repressing pathways involved in membrane transport and lipid metabolism. Mechanistically, DDX5 binds to and drives the translation of CDC42 transcripts, and CDC42 promotes POU2F3 expression to allow for Tuft cell specification. This pathway is parallel to the previously discovered interleukin-13 circuit implicated in Tuft cell hyperplasia, but both pathways drive Tuft cell specification through augmenting POU2F3 expression in secretory intestinal progenitors. Overall, we found that the RNA binding protein DDX5 directs Tuft cell specification and function to regulate the microbial composition and disease susceptibility of the intestine. Further investigation into the DDX5 CDC42 POU2F3 circuit and the DDX5-mediated regulation of intestinal epithelial cell function may elucidate therapeutic targets for combating ileal inflammation and colonic cancers.